and thank you very much to the Aberrant Institute of Research for kindly hosting this talk and for allowing me to share my work with you. And thank you for the kind introduction. I will start where Joan left, which is trying to give you an overview of how people see the evolution of, that's perfect. The evolution of what they call, it is called genomic medicine. And they understand by this that part of medicine which depends on the genomic research. As you have noticed, we have a problem with the space between words, so they are going to try to load the presentation in another computer. In the meantime, I will continue with my presentation. So, about this picture, what do we have in here? What we have in here is, according to these authors, how genomic medicine is supposed to evolve in the next years. They have devised these 5 steps: understanding genome structure, genome biology, disease biology, medical science, and finally, what they call improving healthcare effectiveness. That is what they call research has arrived to the bedside. Research has really arrived to the patient. Of course, I repeat, this refers to genomic medicine, a part of medicine. Now, this is the time scale. The time scale they have plotted here. So, by the end of the 20th century, was marked by the genome, by the sequencing of the human genome, and people were focusing on understanding genome structure. Now, very little research in understanding genome biology. We didn't have yet enough data, and even less in understanding disease biology. Now, this progressed, and in the last 4 years, 6 years, the nucleus of research started to move towards understanding genome biology. Still between these 2 areas, structure and biology, but already moving to the understanding of disease biology, understood in genomic terms, in massive terms, not in specific systems of which you had already a lot of experience. And we started to arrive in here. Now, what do they see? What they do imagine for the next 10 years? So this will continue moving. Now we are reaching a very exciting point. We are putting together all this massive amount of data, and we are trying to understand disease biology in a very general and very broad level, broad in the sense that across the biological hierarchy from molecular level to patient level. And finally, we expect beyond 2020, and I hope I will be there to see it and hopefully to contribute to it, we will be reaching the moment in which all this effort you have done, all this effort that we also have done, that many people have done, will crystallize into something which was kind of a dream some years before. We will advance massively medical science, we or you. So, and finally, a thing which worries us, and it worries also governing bodies that fund research, we will reach the moment in which we will be able to improve healthcare effectiveness. So, a quick switch. I will continue talking because so the problem in here, you are not seeing the problem in here. This is an idealized scheme. You may agree with it or not, but the problem is the amount of information, the huge amount of information which is handled there. We can no longer do things manually. We had to use computers. And I will show you that there are 2 levels. One is the genome level, right? In the genome level, we will have results from RNA-seq experiments. That's perfect. We will have results from massive experiments, sequencing, proteomics, expression arrays, structural genomics. This is the molecular level. Now, the one to which I am closer, at the patient level, at the other side of the coin, We have plenty of data from patient samples, disease characterization, treatment results, and all of this is massive. And actually so massive that it happens something which didn't happen before. You can no longer understand the results of these experiments, without the use of specialized computational tools. You can no longer take the results of a sequencing experiment and just by hand interpret it. You have to use specialized bioinformatics there just to help you to interpret the first experimental results. I do not mean to get a synthesis of all of these, just to interpret your experiment. Now, the same may happen at the other end, and this huge amount of information, this growing complexity, is leading to the creation or to the appearance of a research field that it is called translational bioinformatics. And the aim of this area, which is ill-defined, is not like, I would say, biochemistry, in which people have been working for many years. Is an ill-defined research area in which the broad goal is to develop tools, computational tools, for produce clinically valuable results. Disease prediction, prevention, diagnosing, understanding, treatment, etc., etc. Our main research, we like to believe, lies somewhere within this area. Now, this is a short outline of the talk. Most of our research, as I mentioned already, revolves around the understanding of the molecular basis of diseases, and there will be three parts to my talk. First, understanding protein mutations and see whether we can predict them on either, or understand them either on a very precise basis for very reduced systems, or in a massive way for hundreds of mutations, polymorphisms, thousands of them, hundreds of thousands, etc. Then we will change our way of looking at biology. We will think in a phenomenon which is of growing importance, alternative splicing, and I will show you our work on the characterization of alternative splicing. For now, unfortunately, only in health, but we are starting to work in the understanding of alternative splicing in disease. And finally, part of the name of the title, part of the word of the title, I will show you the effort we are making towards jumping one step in the biological hierarchy, in going from the molecular level, where most of our work is done, towards the cell level. The goal is that can we meet, both you and people working in this research, in some way, at some point, which is not only the molecular level. Can we help you? Can we understand together with you? Can we learn from you the compositional basis, the composition of tissues at the cell level in health and in disease? You know more than us. We want to learn in order to automatize some understanding, some prediction of diseases. Now, as I have already said, my group is essentially or only a computational group. We do bioinformatics and computational biology. These are slightly similar, slightly different research areas, and all the results I'm going to show you have been produced computationally. I will not work through any methodology on my talk. Nonetheless, I wanted to let you know, or to give you a flavor of the range of tools we have used, and which are listed in here more or less orderly. We use sequence level analysis, simple sequence comparisons, of which I am sure you have done many of them, structural or multiple sequence alignment, etc., etc. Structural level analysis, which means structure comparisons, analysis of interactions with interatomic interactions, interprotein interactions. I would like to specifically mention molecular dynamic simulations for one reason. Although it is put on the same level as the other techniques, this is extremely expensive in computational terms, and it is the kind of experiment we do. This is a genuine experiment. For example, different from bioinformatics, where you just gather data and try to make some synthesis. Here we simulate the system. Well, we look at the pathway using bioinformatical tools, biological pathways, and other tools, of which I would mention the use of artificial intelligence tools that serve us for one purpose, one very important purpose, the purpose of synthesizing these huge amounts of information and transforming them into sample models that have in them some knowledge and some predictive power. Now, so much for this. Let's re-understand. I apologize. I have a little, it's warm. So this is the first part of the talk. Just to set the stage, we are looking at proteins. We are looking at mutations that affect proteins. We want to understand them, and we want to be able to predict the damage of a mutation, the damage caused by a mutation, without knowing anything more. That is, we are given a mutation, and we can tell after processing some information whether this is going to be pathological or not with some error margin. So this is the cycle, the knowledge cycle we have. We start with some representative mutations. We process them manually. We understand damage with this manually processing, and at some point we get some principles. We know something about the behavior of the system, about the behavior of these pathological mutations. Then we can build a computational model. Subsequently, we use this computational model to score many mutations. As I said, this can be ultra fast. This doesn't mean ultra accurate, but yes, ultra fast. We can score mutations, we can prioritize them, and then we can tell you, okay, from this list of 2,000 SNPs, the most likely to lead to disease or to be related to disease are these 20. Then you say, okay, I'll do some experimental study. We can also go backwards and reanalyze them. And with this and going backwards, we can improve on our model. And this is a cycle that never ends. Now, as I said, I will skip all the bioinformatics methodology, and I will illustrate mostly things with some examples. The first thing, when we think about protein damage, we think about, in general, three aspects. The damage to protein stability. Is it stable, or will it degrade quickly, or will it unfold very much, etc. Then function, functional interactions it has with other proteins or substrates. And finally, something which is forgotten in many cases is whether damage to a protein or a change to a protein caused by a mutation alters its relationship with the other molecules in the cell, causing the appearance of pathological aggregates, for example. Now, it is very easy to say this, but how do we understand this? The best way to do it is when we have a protein structure of experimental origin available. This is ideal because then we can look at it and we can check the interactions at the mutation site. We can do some computations. We can do some visual interpretation. And although this is approximate, we can get a feeling of what this mutation is doing to this site. We can do the same looking at protein interactions. Here is the interaction between an effector, an epigenetic regulator, and a histone tail. We do not see the remaining of the histone. But then we can think, if something happening here, if we replace a glycine by a tryptophan, which is a bulky side chain, something is going to happen to this interaction. So having a structure allows us to interpret this step. And finally, although at a very coarse-grained level, we can start to think about what happens in the cell milieu, right? I will illustrate with a few examples how one can look at the impact of mutations, of pathological mutations on protein stability. These are examples, some taken from our work, some obtained on purpose for this talk. And what I want to show is, for a set of couple of mutations, how people may look at them. Say, for example, this case, this disease, feline congenital erythropoietic porphyria. This is a feline disease. People use this model, the feline model, to understand porphyria. I am not an expert, I confess it, but this is just from the paper. Now, the protein originating the disease or associated to the disease, uroporphyrinogen 3 synthase. The mutations, these people have identified in here as causing the disease. 2 mutations, a serine replaced by a phenylalanine and a glycine replaced by a serine. Now, what they have done, what these authors have done, is to build a homology model, that is to build a structural model for the mutant protein and to compare what happens in the native structure, which is shown in gray, with what happens in the mutant. So, in summary, what they see is that some interactions are disrupted, part of the normal contacts in the protein are broken, and this would explain, this allows them to rationalize the activity decrease measured in this enzyme, which is nearly 80%. Sometimes it is easier. Like, for example, in fibrillin, this mutation, a cysteine is replaced by an arginine. In Marfan syndrome, here is the cysteine, which is mutated. You observe that it forms a disulfide bridge. An immediate interpretation when this is broken, this cysteine is eliminated, is breakage of the disulfide bridge, and a lot of literature is telling us that the protein immediately becomes unstable. It may form non or less stable aggregates when it is a structural protein, which could be the case in here. We can refine this analysis. We can also compute the electrostatic potential. This we have done in here, I have done it in here. This is very approximate, just to give you a flavor. This is the wild type protein, this is the electrostatic potential, just in very few words. This tells you how the protein likes to interact with other proteins, with other substrates in the cell. I have boxed in yellow in here the mutation locus, right? Here is the cysteine, which is mutated to arginine. Now in here, what I have built is a model of the mutant, right? We have replaced the cysteine by an arginine. This is very simplified. I do not claim this is exact, just gives you a flavor, a qualitative view. And what you see is that here we have red and white, this is the potential, but now suddenly we have in here a blue, the red is less strong. What this is telling us is that because of a change in the electrostatic properties of the surface of the protein, this protein may want to have different interactions, or the normal interactions it has in the wild type state may change or may become weakened. This is, as I said, only an illustration. So, remember on our knowledge cycle, we are in here, analysis of these examples, of many other examples, sorry, have led us to a series of rules that define damage caused by disease-causing mutations. They are essentially rules that tell us about structural disruption, loss of secondary structure, accessibility, things happen in the core of the protein, change in surface properties, loss of disulfide bridges. Now, in here, you would tell me, and I am sure you would tell me, okay, let's continue, Javier. We can already build a model. And this is true. This is true, but there's a limitation. The limitation, I am sure you are very aware of it, is the fact that to compute these properties, to be able to build this model, we need protein structure, right? And this is not a very frequent situation. This happens, of course, but in many cases, mutations, disease-causing mutations, or mutations associated to disease happen in those parts of the protein which have not been solved experimentally. So, can or can we not, cannot we continue with our cycle? We can. I'm sorry, I sound like Obama. Yes, we can. We can do it if we do one thing. If we are ready to lose some accuracy. If we are ready to say, okay, we do not have protein structure, but we can replace by some sequence properties. We can replace secondary structure and accessibility by prediction, sequence-based prediction. They are very good. We are reaching now 75%, 80% accuracy in their prediction. Now, this is more difficult now for surface properties and disulfide bridges, which are intrinsically structural properties. But we can replace them using amino acid sites. For example, if something small is replaced by something big, we may think that if it happens in an exposed region, this is going to be bad. The same if we change the hydrophobicity properties. And most of all, the very best property, almost as good or even better than knowing structure, conservation degree. And let me picture it for you. Now, this is the multiple sequence alignment for this gene, tumor necrosis. I won't go through all of it. For this gene, which is associated to these disease traps. And here we have an alignment. This is not a perfect alignment, right? But this is a reasonably good alignment obtained with Prank, which is what I would say a very recent software, and manually modified. We can do more modifications. I just focus in this region. Why in this region? Because when this cysteine is replaced, cysteine 99, the one in here, is replaced by a serine, it is associated, or it is said to be causing the disease. Here's the human protein. What do we learn from this multiple sequence alignment? I am sure you know, but we learn is that this is conserved across all the members of the family, in spite of the sequence divergence, in spite of the structural divergence. So this means that this cysteine has to play an important role. Otherwise, it shouldn't be so conserved. This is not completely true, but it's a good approximation. So we can use, instead of many structural properties, we can lose without lack of accuracy in the prediction, conservation instead of these structural properties. And now we can go. We can finally build our computational-based method. I apologize, just 1 second, which I show in here outline. We have the protein. We have in here the protein. Shown in red is the mutation. What we do is to label the mutation, right? And which labels do we use for this mutation? These numerical labels, which are not something obscure, not something you wouldn't understand. This is just to put numbers to all the properties I have told you before: sequence conservation, structural impact, volume change, hydrophobicity change. And then we feed this vector to this neural network. The neural network is essentially a very smart program. We can train, we can make it learn about disease, and we can afterwards use for prediction. This program, this neural network, is going to tell us two things. If this mutation is either neutral or pathological, and then it comes with an error bar. It is telling us, it will tell us whether this mutation, whether this prediction is reliable or not. Why is this so important? Well, we will rarely use this for one mutation, because for one single mutation, it is usually much better to do all the process manually, to work close together. You can tell me some properties you know about your protein, some proteins you believe, although you don't know about the protein, etc., etc. We can put all this knowledge together. We can build a good model for the mutation. We can tell this probably much more accurate way. But when we are working with, say, for example, you present me with 100 polymorphisms you are interested in, you want me to prioritize them, and you tell me, Javier, characterizing them at a very deep level is going to be very expensive. Just please prioritize them to me. I will not prioritize them according to the prediction, to the pathological character of the mutation. Of course, yes, you are probably interested only in pathological. I will prioritize them according to the error, because you want to spend every euro you have in something which is likely to be valuable. I mean valuable not only economically, of course, understanding, of course, patients are at the very end. But immediately after that, there is the economical consideration and all of these other considerations. Now, This is the performance of the method, about 84% accuracy, overall accuracy, with false positive and false negative rates around 20%. Is this good or not? Well, there are other methods. When we started this work, there were other people working in this, and we have been considering other things. I am sure there are now some more refined methods, but technically, the main principles I have told you about are essentially the same. And people are still working with sequence conservation, some structural properties, etc. Is this perfect? Is this the best thing we can do? And the answer, and I'm sure you are thinking about it, is no, of course. Because this is essentially a discrete prediction. We are only saying pathology or not. We are not telling you the degree of severity. And I am sure you would like to know these informations. None of these methods that I really know is able to provide you with this information. I illustrate this with this work, as I probably could have done with other works. This is the case, I am thinking in the case of congenital erythropoietic porphyria. It is caused by mutations damaging this enzyme. But one thing very interesting found by Tofigueras and colleagues is the fact that if we go upwards the cycle and we look at allosynthetase 2, they have found that gain of function mutations can increase the severity of the disease, right? This we cannot predict. And this is something we would like very much to work on, these kind of things. So, summary of this part. First, we can understand using bioinformatical tools disease-causing mutations. The level of understanding varies depending on whether we have structure or not, but it can be quite good, at least as good as to design experiments based on this. And then we can automate this, and we can massively score mutations, polymorphisms. Future directions, I already told you about them, but one of them is create tool for specific systems. That is, we have developed our method using thousands of mutations from different proteins associated to different diseases. This gives you an average behavior. We had an idea, and a friend of ours, Alfonso, has tried it also in protein kinases, that it really becomes better, the method, when you train it on specific properties, on specific proteins. This is not surprising, but this is not a thing that has been done extensively. We would like very much to try to do this. Now, the thing I told you already, can we go beyond a yes or no prediction? Can we say something about the phenotypic spectrum of a disease? We can do this. We probably could starting to do this either in a blind way, which I won't mention now, or thinking in terms of pathway, a system view. And then, of course, the dream of everybody is, could we apply part of this to the understanding of complex diseases? Well, first third of the talk, don't despair, we are going fast. So we had a part of the picture. This is understanding of the molecular basis of disease. We have looked at proteins. Now, the problem is more complex, as you know. I have put in here 3 factors: damage to proteins, damage to promoters, and damage to alternative splicing. Right? We became very interested in alternative splicing a few years ago because of the reasons I'm going to tell you. Excuse me. Because it was told that in many diseases, in an unknown number of diseases, not known number of diseases, mutations affecting the alternative splicing pattern were able or would be leading to the disease. That is, we were no longer thinking in terms of protein damage, we were thinking in terms of damage to the alternative splicing pattern. Right? And this is the second third of the talk. I will be talking about alternative splicing in health and almost in disease, but not yet. So these mutations may have different effects. Here is the gene expression scheme of whatever gene. The gene is switching between inactive state and an active state. It may produce mRNA and then a protein. What alternative splicing does, and I am sure you know, is to add a new branch, a new product which is expressed by this gene, right? What happens in the diseases we know. And this is something we have been reviewing many cases. You may essentially, due to a mutation, express more main isoform than expected. So the ratio is imbalanced towards main isoform. The reverse may happen. You are now expressing, you are carrier of a mutation that leads to you to overexpressing the alternative isoform instead of the main isoform. You may express aberrant isoforms that just quickly become degraded, but look at the subtle effect in here. It may not be this guy. It is the fact that what goes in here doesn't go in here. So these have regulatory properties. And finally, we can finish the arrow, the red arrow. This is not degraded or not completely degraded and leads to aberrant proteins that can be damaging or not. So we wanted to reproduce, so these are just statistics that we draw from a review of diseases, of alternative splicing caused diseases. We found that those arising from defective proteins are 44%. Those related to amount deviations, you remember, either by generating aberrant mRNAs or altering the isoform ratio, are about 54%. And then we have a 2% which are due to the fact that AS, alternative splicing machinery, fails. And this is very bad because alternative splicing machinery, although there are not many genes involved in it, controls splicing of all genes, and it is believed that between 90 and 100% of human genes do have alternative splicing. In any case, we became interested in understanding mutations that affected alternative splicing, and we had here our nice knowledge cycle, but we had a problem, an important problem. And the problem is that, different from the case of mutations affecting proteins, is that we do not know very much about alternative splicing at the protein level, its effect, its impact on protein. We wanted to focus mostly on the alternative splicing that went all the way through to the protein level. Right? We have no idea of what many isoforms do. If you go through the literature, you will see has a regulatory effect, may work as a negative dominant, etc., etc., etc. So we thought, okay, the first thing to do is to understand normal alternative splicing. And then we will go, or we will be able to go to the understanding and prediction of alternative splicing damage. This took us more time than we expected. Now, at the protein level, alternative splicing, when you think in terms of the gene structure, may be quite complex. You may lose an exon, you may retain an intron, etc. But at the protein level, actually it is very simple. It only has two kinds of effects. It can either introduce an insertion or a deletion to a main isoform, or it can produce substitutions of sequence between isoforms, that is, stretches that have different sequence properties. When we decided to understand these changes, the problem we faced was, how can we understand them? Can we go through all of them? Can we list all of them? We need a reference. And the reference we used was that of gene duplication. Gene duplications is a well-known, well-characterized phenomenon. Is the fact that at some point in human history or in mouse history or whatever, a gene duplicates and its sequence starts diverging. The divergence, again, is due or is produced by small insertion deletions and substitutions. exactly the kind of phenomena we found for alternative splicing. So we said, okay, let's compare alternative splicing with gene duplication. Let's see what do we get in the case of insertion deletions. This is the size of the chunk we put the size of the insertion deletion. It goes from 0 to whatever. In green, you have alternative splicing values, right? This is frequency. And these are two models of gene duplication. And what we see is very simple. They are different. Insertion deletions in alternative splicing tend to be big, tend to be large. Insertion deletions in the duplication process, followed up by diversions, tend to be very small. Actually, if you look at this is the protein domain size boundary, which means that most of the protein domains are higher than 15, 20, 30 amino acids. Many insertion deletions in alternative splicing are much bigger than this boundary and bigger of the normal domain size. The contrary is the case for gene duplications. Now, can we get some understanding? Can we say, okay, these duplications for alternative splicing are going to do this and that, which in the end is what we want. And this duplication, and these insertions are going to do this, this, and this. Well, we can draw a line in here, and we will say, these, which are smaller than the domain, we will do some modeling. We will try to get the structure. Those insertion deletions that are bigger than this value, that more large than a domain, we can do something very approximate. We can look at the domain structure, see what part is deleted, and because protein function is modular, we can infer in some sense, we can approximately infer what is the functional damage. This is less accurate, of course, but it has value. This, no possible, no way. And why not? Because we do not know. We are left with the problem. We do not know what small insertion deletions in alternative splicing do. Another result I have, which I haven't shown, tells you that they tend to happen at positions different from those found in the well-known gene duplications, which has been modeled structurally thousands of times, etc. And more, the few cases, about 10 cases, for which we have the structure of two isoforms, main and alternative isoform, are telling us one thing, that the structural effect is unexpected. In two ways. It causes some changes that cannot be predicted with present methods. And in a second sense, it tends to produce unstructured or to be related with unstructured regions. Now, this is simpler. We get less quality of information. Sorry. I took this example, as I took the others, because I felt it could be of interest. This is the case of this molecule, two isoforms, a long isoform that presents an insertion at the N-terminal end, no structure available, at least to my knowledge. What could we do to get this information we wanted that we couldn't obtain with comparative model? We can use low resolution prediction methods, like for example, secondary structure prediction. And this is telling us coil, 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 coil with very high reliability. This is coil is the word for something which has a not very well-defined structure. Now, we use a disorder predictor. And this is the insertion right in here. And most, or at least more than half of it, is predicted to be disorder. These, as I said, are approximate predictions. Coarse grain, low resolution. Nonetheless, they are consistent and they may be of some guide. For the larger insertion deletions, the situation is much simpler or more difficult, but functionally more simpler. What you do, what the alternative splicing does, is simply kill a domain right? You have your protein. You have a binding domain, a catalytic domain. What alternative splicing does is, for example, completely killing the catalytic domain. The resulting protein has properties of dominant negatives. Not that we have shown it, not that we have the data. It's just simply because it has the properties, the sequence properties of dominant negative of the main isoform. For substitutions, the same procedure, compare alternative splicing with gene duplications and see what happens. And what happens is the following, again, large difference. What I am showing here, this is the protein sequence. Shown in magenta are the different substitutions. They could be produced either by alternative splicing or by gene duplication. We have measured the maximal distance, that is the number of residues between the two ends, the most remote substitutions. We have normalized it by the size because we are working with proteins of different size. And what do we see? That this maximal distance for alternative splicing, which is shown in green in here, is different from the maximal distance for whole duplications. Now, small values means that the substitutions, that the changes to the protein happen at very precise locations, at very nearby, or very concentrated at specific points of the protein structure. On the contrary, large values, the ones we find for gene duplications, are telling us that substitutions are spread all over the protein sequence. The general inference is that in this case, in alternative splicing, we have changes that are likely to modify function in a very subtle way, in an almost surgical way, while duplications require much more changes along the sequence and are unlikely to give the same effect. Just to illustrate this, please focus on this structure. This is the structure of MAP kinase 9. I have boxed in here the changes between two of its isoforms. They are colored in here, and here, between here and here, we will be comparing gene duplicates. This is, again, the structure of MAP kinase 9, the one in here, but in color, we have the differences with its homologue, MAP kinase 10. And look in here. Changes, substitutions are no longer restricted to the yellow box. They are spread all over the protein. As diversions become bigger, we have 46% of sequence identity only, the amount and the spread of the substitutions becomes bigger. So, to summarize what we have learned about normal alternative splicing, at the protein level, remember, is that we have two gross mechanisms. One is essentially, we can call it quantitative regulation, which would be something like acting at the promoter level or decreasing the normal protein function by generating dominant negative isoforms, or by generating so small proteins that they are essentially functionless. On the other side, we have that alternative splicing, mostly through substitutions, can generate proteins that are variants of the same function, happens at the same or very similar location of the protein functional space. So, and this I will skip, is essentially we studied the relationship between alternative splicing and gene expression, and we arrived to the summary, and the summary is this one. Much alternative splicing at the protein level has a regulatory nature, at least from looking at the sequence properties, and alternative splicing substitutions are on the opposite side, are a qualitative mechanism of changing protein function. Future directions. Our challenge, in which we are already working, the characterization of aberrant isoforms. Those isoforms that have already been related to disease. No longer alternative splicing. We want to know whether they are similar, whether they are dissimilar to this natural alternative splicing. And second, and this is a direct consequence of what we learned from the congenital erythropoietic porphyria, we want to look at alternative splicing no longer for a specific system, no longer for a specific gene. We want to look at it, when possible, for a specific system and within a pathway framework, no longer an isolated protein. So, we are reaching the end. Well, this is the third part of the talk. You have seen that we have made an important effort in devising as good tools as we could for understanding molecular damage. In the case of protein mutations, I believe that we have gone reasonably far with the limitations I told you. In the case of alternative splicing, we are at the very beginning. We know a lot already, but we are at the very beginning. But it could happen, and I took this sentence out of context from this wonderful writer. And yet, if life should refuse to live there, that is, if our models were not good enough. Look at this. These are our models, and this is what they should be. They should have more detail. They should be even better. They should consider things we have not considered. For those of you who are lovers of the church art, this is one of the stained glass from the Catedral de León, a picture that I downloaded this Christmas. So this is the third part, and I promise this is very quick, because I am aware that now we have been already a long time. This is, we want to improve our models. We want to make them better in the direction of meeting you. But not only at the molecular level. Can we go further up? Can we go to the cell level? So this is what I call from molecules to cells. And the idea, for example, would be the one shown in this picture. We have a given gene. Sorry, just 1 second. We have a given gene. It is responsible of the properties of part of the properties of these tissues. It expresses a protein amount P. And we have in here, for the same gene, a mutation carrier, who is expressing 25% alternative splicing, an isoform, of which we will not concern ourselves now. And the net result is we have only 75% of the protein amount. Our question is, are we able to say, okay, this guy is likely to express a disease later on, or this mutation is likely to be related to some kind of disease in the same way as we did for protein damage. The most easiest thing we could have done, and I have to confess that we thought about it, would be just try to scan the literature, try to find mutations that affect amount, try to see which ones lead to disease, and then draw a line, just this single line. Above this line, we have individuals which are that are healthy. Below the line, less quantity. The individuals are more likely to express a disease. This would be very rough, and I am sure you would agree, but this would be a first approximation. However, something happened. Something very important happened, and is that there was an explosion of literature on what I will call the new paradigm of gene expression. Until very recently, until say 10 years, 15 years, I don't know the precise moment, we were thinking, maybe not a priori, but as a consequence of our experimental approach, about the homogeneity in cell composition, in gene expression in cells for the population, for the cell population of a tissue, right? They would be expressing the same amount of mRNA for a given gene, the same amount of protein, and of course, I am sure that if I asked you 10 years ago, is this true? You all would say, no, this is not true. The problem was to show it experimentally in a very clear visual way. And this is what has happened in the past years. It has been shown using fluorescence labeling techniques that indeed there's a cell-to-cell variability. And this could be simply trivial. this could be irrelevant. You could tell me, well, it is also true that we are made of fundamental particles, I don't know, bosons or whatever, but this has no relevance for our research. The important thing that happened 10 years ago, 15 or 5 years ago, is that it was shown that this cell-to-cell variability is biologically relevant. It plays a role in development, it plays a role in stress response, it plays a role in tissue differentiation. Do not ask me how. I am not the expert. But one thing, it seems true. Stochastic or cell-to-cell variability has come to remain between us. So this is a picture from one of the works. This is Chinese ovarian cells, Chinese hamster ovarian cells. Here you have one cell, another cell, another cell, another cell. You can process these images and you can count the mRNAs and you can build nice distributions. The result of all of this for us was the following. We could no longer look at tissues as homogeneous objects. Our ideal of drawing a line and saying this is disease below and this is health above was severely affected. We had to think in different terms. No longer one quantity, but a distribution, as shown in here, distribution of values for the healthy individual, for the mutation carrier, right? A different way of thinking. So we set ourselves, as in the case before, okay, let's first have a model with which we can work, and then afterwards go back to the understanding of disease. So these were our goals: model, describe, and understand. Model gene expression plus alternative splicing to obtain a view like this one. Describe, that is, have the tools to describe this cell-to-cell variability. In the case we are focusing in the impact of alternative splicing mutations. And then finally, get the tissue level view, right? We got, sorry, this simple scheme that has already appeared is at the basis of our work. This is a simple chemical reaction-like scheme. The only things which appear in here are some constants which are of experimental origin. I have to tell you that if we forget about the red part and we focus only in the black part, this has been compared against experiments and this has been shown to work. I will ignore completely all the computation behind, all the mathematics behind. Just trust me on this one. This works at a coarse grain level, so we decided to add this branch. And the results, and we are very near the end. Remember, this was the simple case, no variability, say the original view, complete tissue differences between individuals, right? Either we had this, all the cells with this value, or all the cells with this value. Now, what have we found? We found these situations that we could call similar to this one, gross tissue differences. Either the distributions were nearly overlapping, but each one pointing towards a different end, or very similar to this one, very tight distributions around a mean value, right? And then we had these also very interesting situations. These are situations for different virtual genes. I don't want to lie to you, I don't want to tell you this is trypsinase, whatever, this is whatever, and this is porphyrinogen synthase. No. These are different situations we have generated to test our tools. And what we see is that apart from the simplistic or the more simplistic situation, we have also, and not rarely, a more complex situation in which there's an overlap of values between the product distributions of the healthy individual and the mutation carrier. We can go further. We can check how these vary with the amount of alternative splicing introduced by the mutation, 5%, 10%, up to 50%. And what we see, I just would point one thing, is that at even high percentages of alternative splicing, the overlap, the coincidence between the distributions is high. So we need to go further to get some understanding on what is how much or not so much difference in the distributions and how this relates to tissue properties. So there's a missing link in here that a gap that we just cannot cover alone. So very quickly, summary, we have a quantitative model for tissue populations, which includes, apart from conventional gene expression, alternative splicing. We can describe main isoform variations, and we have that this property varies with genes. Future directions, we want to work with this model in specific problems. Then we are at the end, so we have come back to the beginning again. The research, we are year 2011. We are some way between here. The main lesson we have learned is that to progress, to be able to provide you with sound hypotheses that you find useful, to be able to develop tools that are useful in collaboration with you. We don't want protagonism, and we don't want to say, oh, we cure that or that. We just want to advance in this direction. We need to interact with clinical and biomedical researchers. And finally, these are the people that at any moment have been involved in this work: Santi Béjar, Jordi Morat, and Cassandra Riera, postdoc and two PhD students. These guys did a wonderful work and already left the group. This is one of my collaborators, habitual collaborators, and part of the studies of alternative splicing that I believe were very interesting were done in collaboration with Sarah Teichmann and Christine Vogel. So thank you very much for your attention, and I will be glad to answer any questions.